Now, questions.
Perfect. Thank you so much for that wonderful talk, Dr. Andre. We will now be moving on to the question and answer portion of today's webinar. Again, if you would like to ask a question, please use the Q&A feature, not the chat. Let's get started. First question. It would be great to hear how you go through the Dura. Our lab has had some issues doing that in V1. What is the diameter of the U probes you use?
We use fairly chunky U probes. Around, I think most of these ones we're using were 360 microns, and that's on purpose, just to give it that kind of rigidity. Let's see. Can you guys see my video?
I actually took over a screen share, but you can take over again.
Yeah. Let's see. I can share. I actually brought a handy prop. Let's see. All you have to see is actually my video. Can you see that?
Your webcam? Yes.
Yeah, can you see my webcam?
Yes.
Okay, so I found this in my random collection of Halloweenies, but I thought it looked a lot like a U-probe. Can you guys see it? So it's essentially, imagine this is the U-probe. So this is the part that connects to the head stages, and then this is the length of the U-probe. And imagine this little thing is the guide tube. So if usually what happens, you're going down to the guide tube, and then at some point you start seeing some buckling. So what I do at this point is so we have a NAND system, so it clamps right underneath the head stage base. So I'll release the clamp, and that kind of corrects the alignment. But then you have a U-probe that's kind of sitting loose, just kind of loosely in the clamp. And then what I do is I'll take a pair of fine tweezers, like little forceps, and then I'll pinch it just above the guide tube. Not pinch hard, so I wouldn't recommend this if you don't have good hands. If you have kind of a delicate touch, you can grab it with forceps. And then I take my finger at the top. Remember, so the clamp of the microdrive is still in place, so that's gonna act as a stop. And then what I do is I'll guide the U-probe, kind of push down at the top, and then use the forceps to kind of pull it in, if that makes sense. And then you'll kind of hear a pop and kind of a rush of signal once that goes in. And this way you have some control over how much force you're actually inserting. And then because you still have the clamp there, it'll stop at the same depth. And then you can resecure the clamp and just continue. So it's that little bit of manual force that sometimes helps just to get through if you get through one of those really rubbery parts of the Dura.
Perfect. That was great. Next question. What was the rationale for pulsing the laser and limiting the duration of the stimulation to 250 milliseconds? Have you explored more uniform and long duration laser stimulation?
Yeah, so one reason we use the pulse is that we do need a lot of light, and you don't have to worry about heating the tissue. And the other thing is that it seems like you have to check your particular opsin. So this channelwood opsin, at some point it kind of inactivates. So if by having it pulse, you give the channels a time to kind of reconfigure themselves to their baseline state, so they're able to open again. And even with at 35 hertz, what became clear is that even with each subsequent laser pulse, you actually get fewer and fewer spikes each time, and that seems to refresh like in the intertrial interval. So you want to be careful with things like heating. So you don't want too much light. And the other time you want to look at the actual channel kinetics of what you're using.
Okay. Next question. Did you evaluate the impact of the electrode implant array on the tissue?
No. So the closest thing we've done is, no, not really. We have done a biopsy for the GTHR2, but that was mostly to see whether or not the virus expressed. But it was hard to tell actually the impact it has on, like, you're talking about tissue damage. I'm sure it's having some damage.
Okay. One attendee notes, great talk. What role do you think synchronization between light evoked and stimulus triggered firing plays in the facilitated behavioral readout in near versus far settings?
Well, it's not quite synchronous. So the laser comes on first, and then the stimulus will come on at that conduction delay. So the actual laser and the visual stimulus are synchronized, but the actual response in V1 is a little bit asynchronous. I'm not sure if it's the synchrony itself that makes a difference. But I've never actually looked at it. It's a great question.
Okay. You mentioned optoelectric artifacts during your talk. How did you ensure that the effects you saw were not due to these artifacts, at least in part?
So that effect is usually in the LFPs. It's a low frequency thing. You won't see it in the spikes. Usually gets filtered out of there. Also, it's something that you want to play with at the start of your experiments once you kind of figure out the settings to see like what's that threshold where you actually see the artifacts.
Okay.
So we would just titrate down the laser, the power of the laser until we get something that we're sure we're not getting any artifacts. But either way, at this point, I still wouldn't analyze the LFPs in this because you can still see something, and then you're not sure if it's real or not. So.
Okay, thank you. Next question. Did you look at delays of activation of different neuronal populations in order to differentiate intracolumnar versus intercolumnar?
What do you mean? So let me get more clarification.
I can repeat it, or we can ask the attendee to kind of expand on that.
Reword that.
Sure. Yeah, so if you're the attendee that asked that question, if you could just expand on that a little bit and we'll come back to you. Next question. Have you tried ramping your light stimulus? We find that with square wave pulses, the effect of the light diminishes. We have been ramping on over a few seconds.
No, I haven't. That's something I've been meaning to try actually. Mostly with one of the other options that I think sometimes you get rebound effects with some of the options, and I think the ramping definitely diminishes that. But no, I haven't tried it yet. It's good to know it works.
Okay. What are the latencies after activation of CRH or CHR2 and the inhibition option?
So the CHR2 is actually pretty quick, and it's much easier to notice that one. So we can get activation within like, I think when we did the measurements with the little ruler, we would use like 2 to 3 milliseconds as considered direct, even though that's a little bit permissive. For GTCR2, it's a little bit more difficult. It definitely seems to build up. It's a little bit slower. So it takes a longer time to notice that drop in firing rates. And then the channel itself takes a long time to close. So it actually stays open for hundreds of milliseconds afterwards. So as long as you know this, you can kind of tailor your experiment.
Okay. What do you hypothesize is the mechanism by which higher order visual areas ignore V1 activity when it comes to decision making for the behavioral perception task?
Yeah, that's a great question. I was honestly really surprised when I saw like how much activation we're actually, like how much signal we're injecting in there. And they don't, it could be that, here's my hypothesis. I think if we train the animals to detect the activation, that signal, they could learn to use that signal. However, because we didn't explicitly train them to be aware of the signal we're injecting, I think they just focused more on the stimulus activity. And the rest, and the other signal that we're injecting in there was considered noise.
Okay. What if one were interested in LFP?
LFP and optogenetics?
Yes.
You could do it maybe if you're interested in the LFP after. It seems to be, it's pretty much just when the light is on, when the light is off. So if you use continuous light, you really should just see the artifact at the light onset and offset. So you might be able to just cut out those little portions and look at the LFP during the light is on. It's really just like the transient changes.
Okay.
Or you could look at the time period before or after the light is on, because there are network effects that happen long term. Or you could look at a distal site. You could just move apart the light source and the fiber optic. Like I showed you, like 300 microns is enough.
Okay, great. Have you tried to use tapered fibers rather than adjusting the angle of your fiber?
So we also penetrate the dura with the fiber. So the fiber itself it's pretty blunt at the tip, but it's inside like a cannula that's beveled. So you're talking about tapered fibers kind of get more light out of them. No, I think someone in the lab tried to make those once, but we have not really gone that far. We find that just like a 200 micron fiber, this way seems to shine enough light for us to get our effects. But yeah, the tapered fibers definitely might get you more light.
Okay. Is V1 the only network involved in this perceptual decision?
No, of course not. So the interesting about V1 is that you need V1 for normal vision. But from what human studies have shown is that V1, you're not really conscious of what happens in V1. But you need V1 to kind of send that information to higher areas that eventually become closer, more related to the actual perception. So this is very early, like how that very important information in V1 kind of gets transmitted down, but there's a lot more steps along the way that need to be addressed.
What is the power output range you use for stimulation?
I think it varied somewhat. I think on average we measured somewhere between like 5 and 15 milliwatts per millimeter squared at the end of the fiber.
Okay. One attendee notes, thank you for the interesting talk. What total volume of virus do you inject, and have you any experience of using retrograde AAVs? And if so, are there any special considerations you need to take into account when using these viruses?
I've never tried the retrograde AAVs. Those sound really cool, but I have no experience with them. Most of the stuff we've done is actually lentivirus, mostly because it's a little bit more specific towards excitatory cell expression, which is we just wanted like some part of the network, not to get everything. So the volume of virus, we would do 5 microliters per column, and then it depended, but sometimes we needed to inject some more. So then we do about so each column is 5 times either like 6 or 8 up to 11 other columns. So quite a bit total, but it was mostly just to have like a large enough a target site essentially. Because even in that 4 to 6 week period between like when you inject and when you come back, even if you do everything to make sure that grid is in the exact same spot, things still kind of move around a bit. So you're still going to be fishing a little bit. So you just want a large enough target area.
Okay. Do optogenetic stimulation change the orientation tuning curve, e.g. how wide the curve is, or contrast response function, e.g. change the saturation point or steepness?
We didn't, for that particular study, we didn't actually calculate contrast response functions for the cells. We did it for the inhibitory one. As far as I can tell, it seemed like the stimulus responses were fairly the same. We didn't also, we were always presenting the same orientation of the cells. You could probably do some kind of like adaptive experiment with it, but I'm not sure what the results would be.
Okay. Next question. Regarding your experiments inhibiting excitatory neurons and recording from a distal site, what is the hypothesis regarding the network effects that cause some neurons to decrease activity, but others have different heterogeneous responses? And how do these mixed changes in neural activity explain the behavioral effects?
Yeah, so I kind of blew through the end of that really fast. So I think what's happening, so you have two, let's say you have 2 columns that are side by side. Most of there's a lot of horizontal projections that pass through layer 3. So I think what's happening is that you have the suppressive effects are due to mostly just a lack of input from the local network that usually act to, I guess, increase stimulus responses. And then what happens is as you go kind of deeper in that column, so layer 2, 3 is going to receive that direct input from the suppressed column. As you go further down, you start involving more interneurons. And then you start, once you have an interneuron involved, it kind of changes the way that cell is related to its lateral network. So you might have some cells that are actually being suppressed by the local network at some contrast, and other cells that are being so the ones that actually show the facilitation actually don't show much response at the low contrast. So it seems like at high contrast, they're suppressed by the low contrast, just because when you suppress the column, actually it activates. So perceptually, I think what's happening is that you have some kind of heterogeneity between the two columns, or between like adjacent sites in V1. So you have some that are more strongly connected, I guess the horizontal projection, and other ones where it's a little bit more heterogeneous, where you might actually have stronger interneuron connections. We tried to figure out whether or not it was a proximity to like a pineal center, where orientation preference kind of changes more rapidly in terms of just physical topography of V1. And that didn't seem to be the case, but it could be you might need like a slightly more refined measurement of this. So anyway, the idea is that you just have the lateral connection between the networks are slightly different, and then the perceptual effect you're going to elicit is going to be a summation of those two things. So you're going to have, if you have a kind of a very heterogeneous local network and a uniform suppression, you might not see any behavioral change. Whereas if you happen to be in a patch of network that's more strongly connected, and you just see like a spread of inhibition predominantly, then it might be easier to see that the behavioral suppression. The other thing to notice is that where we see behavioral effects is really at low stimulus contrast. So you want to have like a not subliminal, but like a very hard to see stimulus, and then you're more likely to see the impact that you're having with the optogenetic manipulations.
Okay, thank you. Next question. After laser lighting, did you try to distinguish direct FR effect versus effects that would have occurred later and possibly coming from the population which was activated first, i.e. not directly due to light in the other column, but from the same column?
This is for the suppressive experiment?
I'm not sure.
Well, for the first one, for the excitatory experiment, we took everything, whether it was direct or indirect, assuming that it was just the sum population activity that was actually mediating this. In the suppressive experiment, it was all indirect effects, because if we didn't, once you get away from the light, you don't see any direct effects on any cell in the column. You only see these funny heterogeneous effects that are only evident when there's a stimulus present. So you're activating the local network. I don't know if that answers it. Follow up if that made no sense.
Okay, perfect. Did you always inject virus in a single session? And was this one long anesthetized session? And did you ever do repeat injections on multiple days, or would the animals develop immunity to the lentivirus, making subsequent injections less effective?
So the immunity thing seems to be more of an issue with the AAVs. I don't think I've seen that with the lenti. We have done multiple injections at different days. We do it awake, actually. So we just, it's not different than a recording session. So we just, instead of, we can either have the end of the task, or they can just watch movies. And then we'll do, I think total in the whole day, we can only do about 20 microliters at best. So in order to get more, if you want to do anything more than what I should like 4 columns in our setup, we probably have to do on multiple days. But I never saw less expression with the subsequent injections. It always seemed fine with the lenti.
Okay. You used 35 hertz laser stimulation. Have you tried to match the stimulation frequency to the intrinsic gamma band recorded during the contrast discrimination task without stimulation, say 40 or 50 hertz?
Yeah, so the 35 hertz was chosen was mostly because of this particular channel rhodopsin. It wasn't like Cheetah or Cheap, which can follow faster rates. This one, when we did some preliminary experiments, we found that 35 was about as fast as we could go without losing spikes, essentially. It's like where the channel couldn't keep up fast enough. I would like to use some faster channels, though.
Okay. One attendee notes, thank you for the interesting talk. Are these single units? Also, what effect would you expect in the mouse visual cortex, which doesn't have columnary structure, quotes, near and type 1?
Yeah, so, I forgot the first part of the question, but the part of it, yeah.
Are these single units?
Oh, single units. It was a mix. So we combined single unit and multi-unit. Usually what I'll do is I'll I'll pull out the single units just to make sure that the effect is there, and then I don't mind including multi-unit as well. With the mouse, it was interesting. Actually, kept meaning to find that paper. It was published, I think, right before our Nature Communications paper came out. There was a study that did something very similar in mice, where they were using 2 photons to activate single units. And then what they do is they would simultaneously, well, close to simultaneously activate units that were either of a similar orientation preference or different orientation preference. And then they had the mouse detect whether or not they saw something. And what they found was that, it was quite surprising. It's a similar principle in mouse, even though the architecture is different. They found that if they could simultaneously activate similarly tuned units, the mouse would detect it. Whereas if you had the same number of units simultaneously active that had a more heterogeneous tuning, the mouse would not see it. So the principle is the same, but the architecture is different between monkeys and mice.
Okay. How long does it take you to inject 1 column with 5 microliters?
It's extremely slow the way we do it. 1 column, let me think. Well, an hour, more than an hour, but an hour and 2 hours, somewhere in there. It's very slow. We also leave a lot of time in between the injection, so we'll put the needle in place, wait 30 minutes just to allow everything to kind of settle down, and then we'll inject, and then we'll wait 5 minutes at least. I think the first time we'll wait even more, and then we'll retract a little bit, wait for things to settle, and then inject, and then wait again. So the idea is that you kind of like injecting into a jelly mold. You want to just go slowly. You don't want to like move everything. You don't want to drag the virus back up as you retract. So everything is extremely slow.
Okay. I think that was actually our last question. But if we didn't get to your question, we will try and contact you after today's webinar to answer that. So thank you again, Dr. Andre, for being here today. And thank you to everyone who attended. This does conclude our Advancing Research with Linear Probes webinar series. Thank you to everyone who participated, and all our speakers, and if any attendees were at the previous webinars. You can watch all these previous webinars on demand at plexon.com, and we'll see you next time. So thank you, everybody.
Thank you.
Thank you.